I present the R bioconductor package, nearBynding, a pipeline that incorporates protein binding sites and RNA structure information to discern the local structure of protein-bound RNA. RNA binding proteins select binding sites based on both structure and sequence, but motifs ascribed to RBPs are often insufficient for explaining a large proportion of binding occurrences. Current state-of-the-art algorithms that incorporate RNA structure into predictions of RBP binding motifs use only sequence-based RNA structure prediction and do not accommodate experimentally derived RNA structure information, which can offer insights into RNA structure that nucleotide sequence alone cannot. The motivation for nearBynding is threefold. First, to visualize RNA structure at and proximal to RBP binding transcriptome-wide. Second, to provide a flexible scaffold to study RBP binding preferences relative to diverse RNA structure data types. And third, to allow for ClipSeq data analysis without peak calling. Using both sequence-based and experimental RNA structure predictions, and without calling RBP binding peaks, nearBynding is able to recapitulate published RBP structural binding preferences and observe new binding profiles. nearBynding has three inputs. First, aligned reads from a ClipSeq experiment, which detects where a protein of interest has bound to an RNA. Second, a list of expressed transcripts or transcripts of interest. And third, the region of the transcriptome you're interested in, such as 3' UTRs or unspliced transcripts. Optionally, the user can also include experimental RNA structure information, but nearBynding predicts RNA structure in the absence of experimental data. nearBynding concatenates regions of the genome to consider only regions of interest, and then performs cross-correlation between the protein binding and RNA structure information. This cross-correlation calculates where RNA structure occurs relative to protein binding, so we can visualize what types of RNA structure are over or underrepresented upstream, at, and downstream of protein binding. nearBynding is able to recapitulate known binding preferences. For example, nearBynding shows that the protein pus1 prefers to bind RNA downstream of exterior RNA structure context and upstream of stem structures, while the protein stauffin1, perhaps the best characterized double-stranded RNA binding protein, prefers to bind RNA in the stem, hairpin, stem conformation. We used myClipSeq data, which maps M6A modifications, to demonstrate the ability of nearBynding to incorporate experimental RNA structure data into protein binding preferences. RBM15 is an M6A writer. IGF2BP1 and 3 are M6A readers, and FTO is an M6A eraser. nearBynding reveals that the M6A reader and writers co-localize with M6A modifications across the transcriptome, whereas the M6A eraser is depleted at sites of M6A modification, as expected. In conclusion, nearBynding is a highly flexible, novel computational method to discern protein binding preferences to RNA structures. nearBynding is efficient enough to run on a personal computer and is available as an easy-to-use R package on Bioconductor. Do not hesitate to reach out if you have any interest in or questions about nearBynding.